All right. It is exactly 3:30, and I want to be mindful of our speaker's time and everybody's time. So, a little bit about, thank you all for coming. A little bit about our speaker. It is my honor to always introduce our speakers. We have Dr. Suzanne DiSalvo today. She completed her undergrad from the Evergreen State College in Olympia, Washington. And then she went on to complete her PhD from Brown in Rhode Island. Her educational background has really been centered on cellular and molecular biology, and she really looks at microbial systems serving as the central core of her research interest. I found exciting and fascinating because between 2012 and 2013, she was teaching here at Maryville, but she's currently an associate professor at Southern Illinois University at Edwardsville, SIUE for short, where she is an associate professor in biological science since 2020. So I am looking forward to hearing her talk about how to start a microbial threesome. When she had sent me that message, she was like, okay, I might change it. And I was like, no, I think this is really cool. And so looking forward to hearing what she has to say. So welcome, Dr. DiSalvo, virtually to Maryville University.
Well, thank you so much, and thanks for having me. I really wish I could have come in person, but I'm finding that my relationship with time has become increasingly antagonistic lately. So I think I threw together the last few slides here and wanted to add more just a second ago. So we will see how this goes. But I also, yeah, I wanted to say that I was doing adjunct teaching there basically when I moved to St. Louis from Rhode Island. Had like a year of trying to figure out like what my next steps were and was able to teach microbiology at Maryville before then starting my postdoc position. And, now I find myself here. So I will probably get to the threesome aspect of this in a bit, but I'll start out more with basic sort of two-way interactions to say that my sort of broad interest in my research lab is in symbioses and specifically in symbiotic interactions between bacteria and eukaryotic hosts. So I'll talk about a simple eukaryotic system that we use in a bit. But as I use this term symbiosis broadly, so it encompasses a spectrum of potential outcomes, which could include pathogenesis, commensals, or mutualists. So anything from, a parasitic or pathogenic perspective all the way to beneficial mutualist perspective when it comes to the host outcome. Part of the reason that I'm using this umbrella term is that although sometimes infections on eukaryotic hosts from bacteria can be strictly like either beneficial or pathogenic, in many cases, this is context dependent. So the same bacterial association with a eukaryotic host could be neutral or even beneficial in one context, but in another context, such as like the host immune reaction or other aspects of the environment, could make that outcome shift within the host. So it's can be difficult to exactly say where something falls along the spectrum. So I'm going to use this term bacterial symbionts pretty broadly and also interchange that with bacterial infections without necessarily indicating that that infection is bad for the host system. So in general, I've been looking at the interaction between bacteria that infect or associate with eukaryotic hosts, and then how the relationships impact host outcomes and modulate according to particular conditions. So the system that I've been using at SIUE and throughout my postdoc is a interaction between the soil amoeba. So these are called Dictyostelium discoideum. They're actually a social soil-dwelling amoeba system. And we can grow those very easily on nutrient media plates. So we can just culture them on plates and grow them up. And we look at the interaction between these amoebas with specific bacterial species that can actually infect them. So in this picture, this is actually a image, a TEM image or microscope image of a bacterial cell. And this is an amoeba cell. And you can see inside the amoeba, there are actually these bacteria that can reside intracellularly. So to tell you a little bit more about that host system, so as I said, it's called Dictyostelium discoideum. And this is a useful model system because it can be easily infected by a range of bacterial pathogens that are actually relevant to human infections. And so people have used it as this sort of simple single cellular system to understand how bacteria may be infecting a cell like ours, and how they can replicate and what the responses of these single cells are. And essentially, the way that these amoebas function is that they actually eat bacteria as well as a food source. So this is a picture of an amoeba consuming bacteria, which are in green here. So they phagocytose them and engulf them. And then if it's a food source and that bacteria can't infect the amoeba, then the amoeba actually can digest that bacteria and use it, to get energy and divide. So this process of just bacterial consumption and destruction is actually very similar to what some of our innate immune cells do when they're going around our system and finding potential pathogens and destroying them. Uh, but in addition to those sort of model interactions that you can look at, this, of course, is a natural organism, and it exists in soil, and it actually interacts then with a bunch of different bacteria in its normal habitat. And as such, we might be able to see how those like natural ecological interactions between these amoebas and the different bacterial consortium soil might play out. So that's really been my interest, is more understanding the interaction between natural bacterial interactions with these amoeba host systems. Okay, so I'll kind of cut to some chase as far as like the background of various things that we've done. But what I'll focus really on are these natural bacterial symbionts that we found to associate with amoebas. We can basically pull these out of the soil and see that a subset of amoebas are already infected with these bacterial species. The genre name of these species is Paraburkholderia, so that's kind of a mouthful. So I'll probably just use, a generic like symbiont bacteria or paraburk or something. But I'm essentially talking about a subset of particular bacteria that can infect these amoebas and do this naturally. They are genetically diverse. So when we isolate amoebas from the environment and then look at the bacterial symbionts that they are infected with, we find that about 25% of amoeba isolates are infected with these bacteria, and that they actually fall into 3 different bacterial species. So I'm showing that here. So we call these P. agricolaris, P. baumannii, and P. haliella. And then within these different genetic groups, there are multiple strains, so different or different variants, you could consider them, that can affect these amoeba hosts. And each of these individuals, so you'll see numbers associated with these bacterial individuals, but each of them might have different sort of phenotypes, so different traits, different ways that they infect their host, and different outcomes that they might have. So it's a nice system for basically exploring the diversity of these potential interactions and how different genotypes might relate to different host fitness consequences. But then also, because we can grow these things so easily in the lab, we can manipulate them and ask, how does the environmental context or other things play out in changing the outcomes that we see? All right. And so I want to also point out that as faculty at SIUE, we have a master's program here, but we also do predominantly undergraduate research. So I have both undergraduates and master's students in my research lab. We don't have PhD students or postdocs. So really this whole system has been geared towards work that you can do in undergraduate level labs. And I also integrate this a lot of the time in my microbiology lab. So I try to add that to like inquiry-based projects for coursework. So I also want to point out some of the students throughout the years that have been involved in really teasing apart the bacterial symbiont side of the system. And so I have some various pictures up here and I'll show you at the end. But essentially everything that I'm going to talk about today has been generated and contributed to by a set of undergraduate and master's level researchers. So I didn't want to forget to point that out. So ultimately, that's just a little insight that we have a system where we can look at bacterial symbiosis in an amoeba host. But my next bigger question that I'll talk about today is what role might other players have, beyond like an environmental context? So what role might other microbial players have in influencing these interactions? Specifically, I was interested in bacteriophages, which are viruses that target bacterial hosts. And this actually harkens back to my undergraduate days where I actually got into science because I joined a lab that was studying these types of viruses. And so what happens when you add a bacterial virus that can target a bacterial symbiont, so maybe can infect that bacterial symbiont, and you also put it within that host system, how does that virus persist or influence that interaction? All right, so I should just point out that these bacteriophages, so we call them bacteriophages for bacterial eaters, they are highly abundant and diverse. So essentially any bacteria out there would presumably have a virus that could infect it and potentially kill it. So as these like diverse, abundant entities, and as we know, dealing with viral infections in our own lives, they can be really consequential to the bacteria in the environment. So these, and I'll call them phages for short, but phages basically can drive bacterial abundance and population structure within any environment that you're going to find, bacteria and these viruses. And they also drive the evolution of bacteria. But as viruses are biological entities, they can also co-evolve. So you can get this dynamic between, bacteria that can maybe resist a bacterial virus, and then the co-evolution of a new bacterial virus that can infect that resistant bacteria. So you have these rounds of evolution. But as a result of their impact on their bacterial hosts, they can also scale up to, impact the general environment by changing bacterial composition, and also, of course, potentially other organisms, such as hosts that carry bacterial symbionts. So they can be really impactful. And I should also point out, here's their abundance. If you think about just a single drop of seawater, it's estimated that there's 1 times 10 to the 8 phages in that single drop. So just add, 8 zeros to a 1 to get a perspective of that number. And when you think about even our own like gut microbiome, we know that we have a ton of bacterial in our gut, and people are really interested in how those influence, our health and even like our psychology. But it's been a whole nother level to look at the viruses that are also inside of our gut system. And the majority of the viruses that are in our guts are actually bacteriophages. So there's these viruses that are targeting the bacterial population that lives in our gut. So it's thought when you look at just 1 gram of feces, you get about 1 times 10 to the 12 bacteriophages. So they're more abundant really than anything else in our system. So I'll back up a little bit to mention their infection cycle. So what are bacteriophages? I know you're probably pretty familiar, with the general idea of viruses and how they infect cells and, can destroy cells. So I'll talk about this in the perspective of these phages. But basically, initially, when you have a bacteriophage that lands on a cell that's susceptible to it, and phages are pretty cool looking. They often have these intense structures. So that's what you're seeing here is a bacteriophage landing on a bacterial cell. They'll inject their genetic material into that cell. So it could be DNA or RNA, but I'll probably talk more about it as if it's DNA. So they'll inject that genetic material, and then that genetic material will be used as the blueprint to essentially hijack the bacterial cellular machinery to synthesize new bacteriophages and new, phage genomes. So ultimately, you would get a bunch of progeny bacteriophages made inside a single infected bacterial cell. When I say a bunch, it could be hundreds or even thousands of individual viruses that get produced. And then you also have enzymes and that will degrade that bacterial cell to ultimately kill it and lyse it, such that these new bacteriophages escape, and then they can go on to infect new bacteria cells, and you get these rounds of phage replication and amplification and bacterial cell death. So these phage infections can basically just result in, wiping out bacteria numbers. And you can see this like if you look at microbial plates. So here's a plate where you have a lawn of bacteria on it. And then within this lawn, there's a small amount of individual bacteriophages that have been mixed in. And wherever any of these individual phages has hit a bacterial host, it's basically amplified, lysed that cell. Its progeny have infected neighboring cells, lysed those, and that process has continued to the point where you actually see a whole clearance zone. So we call that a lysis plaque on that bacterial lawn. So you can see that those bacteria have been killed in that area. And this is actually really equivalent to what you would see if you were looking at antibiotics that are placed directly on a bacterial lawn. So this is a zone of inhibition for an antibiotic. So you can see that, bacterial death outcome that phages might have. And so because of, that kind of outcome, and the fact that lately we're seeing and going to probably increasingly see more and more bacterial infections that are resistant to antibiotics, there's a big urgency to find alternative treatments to bacterial infections. And I think maybe Andre Durantini talked, and I know he's looking at some of that, and we do some collaboration as well. So you've probably heard about some of this even from him in your seminars. But because of that, drug resistance, this has kind of harkened us back to say, hey, if you look at a zone of clearance from a phage, it looks just like an antibiotic. Can you use these bacteriophages to essentially target bacterial infections and wipe them out? And I want to say that this is not a new idea at all. So this has been around, it predates antibiotics. So some of the initial people that were discovering phages and seeing them as sort of these bacterial toxins, essentially, were suggesting that we could use these to treat bacterial infections, and they were even employed. So phages were isolated from the environment and used to treat a variety of bacterial infections in different circumstances. So back in the early 1900s. But of course, since antibiotics came on the scene, they, for the most part, or most parts of the world, sort of supplanted and disrupted the interest in using phages for this. And there's a couple different reasons for that. For one, I kind of mentioned the evolutionary aspect. So, yes, like phages can target and destroy bacteria, but but the bacteria can still evolve resistance to the phage. So you're still going to have issues with resistance, just like you'll have issues with resistance to antibiotics. But I think probably the bigger limitation that comes into play is the fact that phages are not what we would consider broad spectrum, and that one individual phage that you might find and be able to pull out from the environment really is only going to be able to infect a small subset of bacterial types. So it's sort of akin to what we think about with, different mammalian viruses. We have, SARS-CoV-2, unfortunately, seems to have a broad host range and can infect lots of species, but other viruses like human smallpox was specific just to the human species. So when we think about phages, they kind of have that variety. You have some individual phages that maybe target lots of different strains or even some related species of bacteria, and others that really have these limited ranges. And this is just an example of that when it's one of the phages I'll talk about here, but I called it Balix. And this phage, when I add it to bacterial lawns, you can see this clearance here on some of these bacterial strains. And all of these bacteria here are actually the same species. So you can see how it's only targeting and lysing a subset of individual strains within the species. So that's really problematic if you want to use something as a bacterial treatment, because you have to exactly know what kind of bacterial infection you have and whether or not it's susceptible to, this virus that you might have in stock. There's another issue that comes along the idea of really using phages as a therapeutic, which is the fact that I gave you one possibility, one outcome of phage infection, which is this lytic cell destruction. But another possibility is that phages can actually integrate their genome. So you can see that here, instead of coordinating new phage replication, you actually have the phage genome in red integrating into the bacterial host chromosome and then replicating passively while that bacterial cell divides. So in that case, you could have your bacterial population be just fine, and instead the phage is now part of that bacterial genome, and we call that a prophage. So I should say that some phages, they basically belong to one of two types. One type would be purely lytic. So let's say phage A, it may only be able to do this cycle. While another phage, phage B, may be what we call temperate. You can think of the seasons, a temperate rainforest or something, or forest, in that it has sort of this different seasonality. But these temperate phages have this lysogenic or prophage ability and a lytic ability. So for those phages, they may integrate and then passively replicate, but if a stressor, arises, that phage genome can then excise out of the host genome and establish the lytic cycle. So that might be accomplished during a cell stress event or just spontaneously in a small portion of the population. Nonetheless, if you're using a phage that happens to be able to integrate into the bacterial genome, you're not going to be able to rely on that phage to kill a bacterial population. Um, and even worse, this can happen regardless of whether a phage is just selecting for evolution when it comes to, lytic cell death of bacteria or a lysogenic cycle, but especially when you think about integration of phage genomes into the host bacteria, this can actually endow a bacteria with new traits immediately. So sometimes phages are going to carry along genes that they picked up from other host bacteria in their, history of infection, or, they encode genes that actually protect their host bacteria so other viruses don't kill it because their interests are aligned with this host. So um, this can be really problematic and relevant even to traits that could be important for us. So in many cases, these prophages have been found to be associated with virulence genes. So some genes that encode toxins that actually result in a bacteria being pathogenic in a human situation. So this is just an example of pathogenic strains of E. coli actually carry a toxin, a Shiga toxin, that is associated with a prophage. So certainly that's a really impactful consequence of phage interactions with bacteria. So I think going just back to the fact that we have looked at phage therapy for a long time, there's obvious reasons why this has become a complex issue. And where today with modern molecular biology and other techniques, maybe there's abilities to modify things and really target new functionality to kind of overcome these hurdles in the rise of antibiotic resistance. Okay, so I basically was just saying that phage and bacterial interactions just alone from the purpose of, cell death and prophage integration and of course evolution, you can imagine that there's just many different potential outcomes, and it may not really be easy to predict those unless you're doing lab-based tests to see what will happen. But of course, even more difficult is it to predict what would happen if you're looking at that phage and a particular bacterial target within a host system. Maybe it's a host that you want to treat with a bacteriophage, or you just want to understand how these interactions occur in a natural environment. And you add that to other conditions of the environment. So even though we kind of think about phages and bacteria as simple, relative to multicellular organisms like us, there's still a lot of things that are challenging to predict or understand. So I think there's still a lot to be learned. Well, I hope so, because that's what I'm working on, right? Oh, and then there is a weird, okay. So I'm going to go back here and basically say that given all of this, I wanted to basically use that system that I had, looking at bacterial symbionts in these amoeba hosts and the fact that we already have this diversity and this ease of culturing them on plates, and then throw in phages that target the symbionts within the system and kind of see what kind of outcomes do you get. Okay, so so let me just go back to, oh yeah, that was good, sorry. Moving around here. So let me go back to the system again, because I want to go through exactly how this works in the lab, because that'll become relevant when it comes into the type of experiments that I'll show. So I kind of was like, okay, these amoebas can be infected with bacteria and all of that, but they also have a multicellular cycle. So they're not just single individual cells. That's one part of their life cycle, but they can also go through the social cycle and aggregate into what we call a fruiting body. So for perspective, the way that we work with these things in the lab is that we take these amoeba spores, so we're going to call them spores as like our starting thing, and then we can add them to a nutrient plate with bacterial food. Those amoeba spores will germinate into the vegetative amoebas, which, as I mentioned before, will phagocytose bacteria as a food source, and then they'll divide by binary fission. And then ultimately they'll aggregate when food becomes depleted and amoeba numbers are high. They actually aggregate and form a slug that migrates. And in the case of the lab, this is just on the surface of that agar plate. And then they start to form this differential fruiting body, which I'll show, but in nature, this occurs just kind of on the surface of soil. So they are seeking a location to form this fruiting body, and then they ultimately form this fruiting body. And that consists of a stalk of a few cells that support this globular sorus at the top, where there's hardy spores positioned there. And then it's those spores that we can collect and replate on new nutrient plates to generate these fruiting bodies again. And this whole process from plating amoeba cells to getting fruiting bodies and all of that is just about 4 to 5 days. So it's a nice, quick system to use. Okay, and so I'll skip that. See, even here I moved away things. So as I mentioned that I did actually want to talk about this. So I mentioned that these amoebas can get infected with pathogens, but really what's kind of been fascinating is that during this multicellular transition, even when you work with pathogens that can infect them in lab conditions, they can often clear those. So there's some kind of mechanisms at play during both single cell growth, but also this multicellular transition that can actually clear bacteria from that fruiting body structure. So oftentimes, when you look at those sori contents, they don't have any bacteria associated with them. They're basically germ-free structures. But as I mentioned, this isn't true when it comes to a natural scenario. So we know that there's lots of different bacteria that can interact with our amoeba in its natural habitat. I've studied a couple different ones, but the one that I'm really interested in for today are those Paraburkholderia symbionts. And so those are what I'm going to get into, but those aren't the only ones. And for any of these symbionts, when you find them associated in the natural environment, and then you bring those amoebas and bacteria into the lab setting and do your culturing, they will persist. So you'll still get them in these fruiting bodies and spores, and you can transfer them and maintain infections in the long term. And so with Paraburkholderia, not only do we have all these different genotypes, as I mentioned, but I've also been able to label many of those with red fluorescent proteins. So we can grow these bacteria on plates on their own. They don't have to infect the amoeba to survive. And then we can manipulate them. So we can label them with RFP, which allows us to visualize and track these bacterial infections. And so I'll show you essentially what that looks like from stage to stage. So these are our amoeba cells under a fluorescent microscope in gray here, and then the green bacteria is just food bacteria, so that won't infect them, but the amoebas can eat it. And in red here, this is one of our symbionts that's labeled with RFP, and we've basically exposed an uninfected amoeba with this symbiont by just growing it on the same plate. And you can see that the symbiont starts to accumulate within that cell. And then that persists through the slug formation, which you see here in TEM and confocal microscopy here. So all these individual slug cells, some of them are still infected with these Paraburkholderia symbionts. And then in the final fruiting body structure, you see that individual spores here are still carrying Paraburkholderia cells. And as I mentioned, with all of the different strains that we have, we see these different outcomes. So when we look at just sori or spores that are infected with different symbiont genotypes, they have different patterns of infection. So in this example, you can see that almost all of the spores are infected. All of these amoeba cells are infected, and they're infected at pretty high levels. So there's probably like 20 individual symbiont cells within there. In this picture, you can see that there's still pretty decent levels of infection, but it's lower than the other scenario. And we even have somewhere it might be very low levels of infection or maybe high numbers of infection, but low density of bacterial infection. So there's all these different patterns. And those patterns don't necessarily relate to the host outcome. So one might think that the more infections you see in the host, the worse it would be for the host. That doesn't necessarily play out. So there's other things going on. But regardless of the disconnect between infection patterns, we still see a diversity of host outcomes. So some symbionts really reduce host fitness, so they're acting as pathogens, and these are just really short fruiting bodies, kind of trying to give that indication. But ultimately, you get a lot less amoeba growth and amoeba cells on the plate if that bacteria is hurting the amoeba. And others, don't do anything. So the amoebas look just as healthy and happy carrying one of these paraburcholdaria symbionts as they would without it. And even in some environmental contexts, symbionts can be beneficial. So this is our whole spectrum of potential host outcomes that we can see. All right, so the next thing then is let's integrate the phages into this system and see if we can answer a variety of questions, such as what impact then do phages have on these symbiont infections? Does this impact host fitness? And maybe what happens when you think about long-term evolution? So obviously you want to, maybe use phages to treat human infections or something like that, but that's a much more complicated system. Maybe this serves as a nice microbial system to explore the diversity of outcomes that you can get with phage addition to bacterial infected host organisms. And then ultimately, one thing that we would like to connect is how do specific traits that we can measure when it comes to individual phages or individual symbionts, how do those relate to the efficacy of phage treatment, or what those outcomes will be in the host system, and how maybe does the environmental context also influence these things? So again, we can modify and play around with our culturing system to see if we can get at underlying mechanisms that might allow us potentially to predict what kind of impact we'll get when we do sort of a simulated phage therapy trial. And just in general, I'm talking about phage therapy, but really just to understand these relationships in sort of these ecological contexts. All right, so more student pictures. There's been a bunch of students who have isolated phages when I started at SIUE, and then some of my current students, I'll probably just be able to touch on very quickly some of their stuff, but they are all working on some really interesting phage projects that we're getting some new data on. So I have to, again, thank a bunch of undergraduate and graduate students for the phage collection that we currently have and are working with. Okay, so skipping through some of this animation, but the first thing that we have to do is, we have already our amoeba and our symbiont collection. Let's go out into the soil again and see if we can pull out bacteriophages. So can we find and isolate bacterial viruses that target our symbiont? So we essentially collect soil. We harvest that soil in liquid broth, like we might grow our bacteria. And then you can filter the supernatant of that liquid. The reason we do that is that viruses are very, very small, and you can use these filters where there's pores that only allow very small things to go through, but block any cells from going through. So they'll block any bacterial cells from going through while allowing things small enough, such as viruses, to pass. So you can get these cell-free filtrates, which presumably may hold viruses in there, and then test to see if there are any viruses of interest. So that's this filtration part here. There's these little filters you can attach to a syringe, get a cell-free filtrate, and then what we can do with that filtrate is we can spot it on a bacterial lawn and see if we get a lysis plaque. So that's what we're looking for. If we get a lysis plaque, it's likely coming from a phage that was in our soil sample, and we can then go on to amplify it and work with it. What I've found, and you'll see, is that ultimately it'd be nice to just take soil, resuspend it in media or water, whatever, and then filter it and then get phages from there and spot that filtrate on a lawn and see those lysis plaques. But we don't really get anything when we do that. So what we have to do is we actually take the initial filtrate that we get, then we inoculate it with our bacterial strains where we want to pull phages that target them. So we add all of those bacteria in to that filtrate, and we hope that any virus that's in small abundance will amplify on a host if the host is there. So basically, we're trying to enrich and amplify these bacterial viruses that will target the host that we're using. After we go through a couple days of that, we again filter and then test. And so here's an example of then what we're going to look at on lawns. We're going to basically see, do we see a plaque or not? And if we do see one, how strong, like how clear is that plaque? So it's a qualitative scale we're using. 0, we don't see anything, to 5, a nice clear plaque. And I'll display this in a sort of a tile plot. This is what a plate actually looks like. So here's like a square plate held up to the light, and you can see I've spotted various things onto those lawns. In some cases you see plaques and clearances, in some cases you don't. So that's basically what we're looking at. And I'll say also that we've looked at lots of different soil samples. I started this in 2017 and refined our methodology. It should be easy to find phages, technically. You can find E. coli phages pretty easily going into sewer, but we've really had a lot of struggles and challenges getting phages that target our symbionts. So I think it's getting better now with our methodology, but ultimately, out of all of the different sort of samples we've looked at, I've never been able to get anything from around this area that targets our symbionts. But what we have are some phages from South Carolina soil and Texas soil, and now we're getting a few other phages from different soil. But here's the results of our first sort of screens that worked. You can see that before enrichment, anything that's purple here just means that we didn't get any kind of lysis plaque, so likely no phages. But after enrichment, after presumably amplifying phages that target individual hosts, we get some evidence of lysis plaques. So stars here correspond to the bacteria that we put in the filtrate to try to amplify phages that might be there. And each of these numbers is a different bacterial strain. So you can see in soil from Texas, we had lysis plaques on these two strains. They're both within the same species and, a little bit of clearance, but kind of cloudy lysis plaques on some other bacterial strains. And then in South Carolina soil, we also got evidence of phages, and these were targeting more of the lawns that we tested them on. So the species Bonnia, a few strains of Haliella, and a few strains of Agricolaris. Those are all our different species of symbionts. All right, so once we get these, I won't belabor this too much, but we have to isolate individual phages and make sure that our stocks are pure and not a collection. So we go through a process of sort of purifying out stocks of phages, and then we test those stocks again. So the next thing here is just showing the final kind of phage characterization of those isolates that we pulled from soil. And so the names, if you can see those, we just gave them the names. So we have a phage called Bonzo, Piranha, Balex, Scuba, Bagra, Sugar, and Prozo. And then once we know we have this pure stock of phages, we again spot it on a bunch of different bacterial hosts and see what kind of plaques we get to get a sense of the host range. So what are the different strains within our collection that these individual bacteriophages hit? So Bonzo hits only the strains of Bonnia, so it's only hitting one species. And Piranha is hitting both Bonnia species and Haliella species strains. So it has a broader host range. And then you can see these other phages are kind of hitting different species to different effects. And you can also see here. So these are just lawns of individual bacterial species, the individual phages, and the kind of clearance patterns we get. So one thing you might wonder is why do you see different clearance patterns? And this could relate to whether or not a phage can establish lysogeny or not. So is it purely lytic? If it's not purely lytic, you're more likely to see these cloudy lawns. Another possibility is that bacteria have defenses that they can mount against phages. So it could be a bacterial-induced defense that makes that phage not as effective at killing it. So that's the type of variety that we see. So these are some of the initial phages that I'll talk about. Here's also some electron microscopy images of those phages just overlaid here. So that's our image of Bonzo. It's got this head capsid and tail-like structure. Piranha, sort of just a head capsid here. So these are just different types of phage structures. This is a picture of Balix, scuba here, Bagra, and sugar. So we get a different variety of structural morphology when it comes to each of these phage isolates. And you can just see the scale here. So this is 50 nanometers, and this is why we need an electron microscope to even image these viruses and why they pass so easily through these filters that I mentioned. Okay, so finally we have these phages. Let's add them to the system. This is what we're, ultimately trying to do. So again, what impact do these individual phages have on symbiont infections and host fitness? Sorry, and are outcomes variable? How and why? Can we correlate those with any kind of features? So we can basically take amoebas that are infected with given symbionts, and we're going to use here symbionts that are susceptible to the phage isolates that we're going to try to quote unquote treat them with. And then we can also add in the phage, so some phage filtrate to these populations. Let the mixture of these three different entities grow for a week so that we get fruiting bodies from those amoebas, and then we can ask what are our outcomes. So some of the outcomes that we look at are infection prevalence. So what's the percent of spores within this population that are infected with symbionts? Since we use red fluorescently labeled symbionts, we can look at the percent of spores that are fluorescent. So that's our infection prevalence metric. Another thing we'll do is look at just total spores that are produced on these plates. So we can count individual spores. The more spores, the happier and healthier the amoebas are. And then we can also look at what about phages? So we're looking at plaque forming units. So those individual sort of lysis plaques are a way to count the number of phages. And we can ask what is the total number of plaque forming units in the culture, after that development. Are phages, do they persist in the population? Are they lost? And is persistence, associated with reduction in symbiont infections or anything with host fitness? So we're just looking at those three different outcomes. So phage, symbionts, and host amoeba. All right, so first I just want to say that phages are recoverable from amoeba populations following development. So if you add them in, let the amoeba develop and create fruiting bodies after a week, you harvest all of this and filter and dilute and then count the number of plaques that you get. We see that there's still about, 8 times or 1 times 10 to the 8 bonzo phage in Paraburkholderia bonnia 433 infected cultures. So these numbers are a lot because of all the cross pairings that we do. But basically, this is the symbiont name that's infecting the amoeba population. And then this is the treatment that we've provided. So we have lots of bonzophage at the end, slightly less piranhaphage when we treat with that one. This is a piranha treated 171, so a different symbiont, and then our Balix and scuba phages treating a different symbiont infection. So I know that's kind of a lot to look at, but there's different combinations here. So phages persist, but it depends on the phage and the symbiont context. So phage replication and persistence varies by condition. And likely you might imagine that it's because if the symbiont is really susceptible, you might imagine more phages are going to replicate over time. You're going to get higher numbers. And if maybe the symbiont is not as susceptible or has these phage defense mechanisms, phage numbers will decline. So we see kind of a variety of those effects. Next, we can look at infection prevalence, and these are also confocal micrographs. So you can see the difference here, but this is amoeba spores infected with this symbiont here, pretty high prevalence rates. It's pretty clear that benzophage addition reduces those symbiont infection rates. So this is the plot looking at that percent of infected spores for this particular infection. About 80% of spores are typically infected. When you add benzo or piranha to it, that infection level declines significantly. However, piranha, which can target this 433 and 171, doesn't have any impact on the 171 infections, despite the fact that it will give you a clearance pattern on that bacteria lawn. And then you also see the variation in these other combinations. So Balix and scuba reducing these infections, so this symbiont infection, and then this other symbiont already doesn't infect very well, so we don't see a big difference. So variety, infection prevalence can be reduced, but again, this depends on the combination that we're looking at. And then total spores, again, you're seeing that total spores, this relates to the host impact of the symbiont to begin with. Some of these have no impact on the host fitness. So this is the number of total spores we get from typical uninfected amoeba cultures, and only 171 here. And I didn't put the names here. This is 1045, but only 2 of these symbiont infections really do reduce spore productivity. And we can see that only in one of these contexts do phage treatments alleviate this reduction. So we get, again, just as you've sort of seen for everything, you can see some impact on host fitness with phage treatment, but it depends again on the combination. Okay, so then I'll quickly go through this because I'm kind of running out of time. But the next thing we really wanted to do is, well, what about evolution? Like, how long do these interactions persist? Do they change? Could you get further reduction or further amplification? So we basically just transferred spores that grew up from that initial co-culture of those 3 different things to new plates without new infections or new phage treatments. So we're just doing continuous transfers, which is food bacteria, and then asking what things look like afterwards. And I didn't measure everything each cycle, but this is just that first cycle that you saw for everything. And I guess I'll just cut to the chase. There's a lot to look at, but for some things, so this is 433 symbiont infections, bonzo plaque forming units remain high, so that guy persists over time, and symbiont infection levels, so infected spores, remains low. For piranha, it looks like it's not significant reduction, but it looks like maybe the piranha is not maintaining as well. So I might imagine that more transfers of this, we might lose this phage over time. And the symbiont infection prevalence seems to be increasing. These are just a few replicates, so we don't have significance there. If you look at this other example with Balix and scuba phages, you see that especially for, let me look at like this one. So Balix, plaque forming units, we lose Balix phage in some of our replicates for this infection. So some phages are actually lost, others persist. Again, all of that is just to say that there's variation over time. And then over even longer periods, so we've done this with a subset actually with bonds on 433, we see even more strange patterns emerge. So some lines that are transferred regain high infections and even become virulent. But it's hard to interpret at this stage. So my summary and initial thoughts here are that phages can persist and impact symbiont infections. Symbiont and phage identity does impact efficacy of what we're going to see, but really the question would be what traits underlie this. So it could be that phage replication and virulence is just different according to the individual hosts and phages. So these are the traits that you might be able to correlate back. The other thought that I initially had was that maybe infection characteristics impact whether or not a symbiont is susceptible to a phage. So if you can see a phage lyses a symbiont on a lawn, why can't it reduce that symbiont in the amoeba host? Maybe by being inside the amoeba host, it's protected from the phage. So maybe some symbionts, such as this guy, 171, are really good at getting into the host, staying there and replicating versus kind of going in and out to establish infections. And so maybe that's why piranha doesn't do anything to 171 infections, but does reduce 433 infections. And then similar to like phage traits, maybe defense mechanisms are important. But at this stage, I can't really say and go through. So at this stage, it's kind of difficult to understand what exactly aspect of it is most important without really having more phage and symbiont pairs to look at, and then of doing more, especially like in vitro testing. So I'll say one last thing, which is that we do have more phage isolates now, and a lot of these overlap. So we've got 3 more that overlap in their host range and that they can affect the same symbiont strains. And then we have started to do infections with those, but I'll cut to a quick chase, which is that in vitro, so this is just measuring bacterial liquid growth. In vitro, so that's optical density, so how much a bacterial culture is grown, and this is our control. So that's what we expect over about 10 hours. Our 433 symbiont, just grown in liquid culture without amoebas, increases in density over time. If you treat this with bonzo here, that optical density goes down and is reduced. So the phage is lysing that host as we would expect and kind of killing it off in the liquid culture. But strangely, and this wasn't what I was expecting, these other phages, so including piranha in green here, don't correspond to a decrease in optical density alone. So basically, it may be that just doing these liquid infections and looking at bacterial density could predict what we're going to see in the amoeba system, which makes me think that what more likely is influencing those outcomes is whether or not these symbionts maybe can mount a defense against phages. In the cases where symbionts still grow, we also see a reduction in phage prevalence. So I think the phages are infecting and getting destroyed once they get in the symbiont. So those are very preliminary, but that's what we're going to keep focusing on to try to get to the outcome there. And then the other last thing I'll note is that another part of our project is looking at prophages. So within our own symbiont collection that we have, several of them have evidence bioinformatically that they have integrated prophages, which presumably could be functional. And we know that we can get a functional phage out of one of our symbiont strains. And so we're investigating what impact that prophage might have on that symbiont strain and how that influences the infection in amoeba hosts and maybe competition with other symbionts. So I don't have time to talk about that, but that's a whole nother sort of avenue, is just looking at the impact of prophages in the symbionts that we have. Our Again, thank all of the students. Thank you guys for listening to this. I wish I could see you in person, but I appreciate being able to talk to you here, and also thank the funding that I have for this project from the NSF. And I'll take any questions.
Thank you so much. So if anyone from the floor has a question, please go ahead, unmute yourself, or you can put it in the chat, and then we'll answer that. All right. I see Ellie's hand up. All right. Okay. So if there's no one else, because I know Ellie and Morgan, they are in my advanced topics class. If there are no questions from the general floor, then I want to say thank you all so much for coming to this session. I will stop recording right now. And then if you're in advanced topics, you'll just stay behind and then we'll have our class. So thank you all for coming and enjoy the rest of your day. Except my advanced topics students, but I will wish you a good day later. All right. Oh, if you're in advanced topics, don't put your question in the chat, unmute, and then have a discussion. So please, 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 I'm not going to read the chat if you're advanced topics. All right, perfect. I think we do have our advanced topics people left. Please, Jasa, Madison, Ethan, if you can come on video, and Ibrahim, if you can come on video, that would be great. It's the same rules of class apply, so please, please come on video. All right, Ellie, you can go.
Hi, first of all, thank you for today. My question was, how did you choose the symbionts?
Yeah, so is it for the phage treatment part of it, or for just in general, the ones that I'm interested in?
It was particularly for the first one. There, I saw the chart, and then there was three that you had specifically chosen. I was trying to figure
Yeah.
it out, but I wanted to ask you.
Yeah, and let me go back. So for those, it's really kind of like what we have. So the Paraburkholderia species that we have are all from these collections of the soil, and actually a lot of that happened. So even just like these species and strains here, a lot of that happened at Washington University in the lab that I was a postdoc in, the Queller and Strassman